Hello guys, so today we are going to discuss about the cell cycle and cell division. And most of the students have no clarity about cell fundamental process and their regulators. But I promise by the end of this video you'll get to know the main projection of the cell cycle. So the major part of the cell cycle is divided into two major parts, interphase and M phase. And most of the time cell cycle is in the resting phase, that is G1 phase. Then S phase, after the G1 phase the DNA is completely replicated, and the last phase is G2 phase where the cell continues to grow and verifies that DNA has no mistakes before cell division, and this cell divisions is M phase. As you know that the M phase is a mitosis phase. Why? Because mitosis where the chromosome separates and cytokinesis happen, and you also know that cytokinesis is actually a one cell splits into two daughter one. Okay, so now the regulators are cyclin dependent kinase CDK and cyclin. So each checkpoint has its own cyclin. So the cyclin D works for G1 phase, cyclin E works for intermediate between the G1 and S phase, cyclin A works for S phase, and cyclin B works for G2 to M phase. So I have already made a chart where it clearly shows that each CDK and cyclin how it works for the cell cycle progression. Now we understand how cyclin works. Cyclin act as a switch. Okay, their level rise and fall during the cell cycle. When a specific cyclin bind to the CDK become active and phosphorylate target protein that derive process like DNA replication, mitosis and cell growth. So the simple relationship is CDK are the engines and cyclin are the keys. Without cyclin, CDK cannot function. The cycle begins in G1 phase where the cyclin D binds to CDK4 and 6. This pair helps the cell grow and pass the restriction point, which is the major decision step to continue the cell cycle. Next is to enter the S phase, the cell uses cyclin E with CDK2. This combination tells the cell now start DNA replication. Once the cell is inside the S phase, cyclin A bind to CDK2. Finally, before the cell divides, cyclin B activates the CDK1 in G2 to M transition. This powerful complex triggers mitosis, chromosome condensation, nuclear envelope breakdown and spindle formation. So in summary, we can say that CDK4/6 cyclin D start the cell cycle, and CDK2 cyclin E enters the S phase, and CDK2 cyclin A continues the DNA replication. Together, these CDK cyclin pair ensure the cell cycles